Welcome to Mr. Chalk's Revision Tips. In this video, we will look at if we remember from previous videos, bacteria are prokaryotes, so they lack a nucleus and they're single-celled, but they do go and reproduce very, very quickly. So, when looking at practical activities in science, there are some important keywords that we must focus on. So, control variable is any variable that does not change during the experiment. The dependent variable is the variable that is being measured during the experiment. A hypothesis is what we think will happen as we go and change the dependent variable and the reason why. The independent variable is the variable which we are changing. An anomaly is an observation that differs or is out of sync or out of the pattern to what we would expect. A correlation is the relationship between two variables. An error is a measure of the difference between the measured and calculated value or a true value. And the difference between precision and accuracy, often interchangeable. Measurements that are closer to the known value are said to be accurate, whereas measurements that are close to each other are said to be precise. So, when we're looking at how we're going to set this up, the first thing that we need to do is to set up a Bunsen burner that's going to heat up the air around where we're working that will kill any of the bacteria that's floating around, so you don't get your culture contaminated. We then go and sterilise our inoculating loop by dipping it in ethanol, then burning it off. That's going to kill any bacteria that's already on there. We're then going to go and transfer some bacteria from a bottle or container onto an agar plate, and then we will go and re-sterilise the inoculating loop and seal the agar plate in 4 places using tape. It's important that we still allow some air to get into it, as we don't want to grow bacteria that can grow without oxygen. We can then incubate it at around 25 degrees for 3 days before looking at the results. So, if we have a look at some quick questions, so here's a diagram just summarising the method that we've just looked at. So, what is the name of a piece of equipment A? That's going to be our while loop. What's the name of a piece of equipment B? That's going to be our Petri dish. Why should apparatus A and B be sterilised before they're used? So only the bacteria that we want to grow there. There's no contaminations. Give one reason why the lid should be taped in place securely. Right, we tape it in 4 places so that bacteria that can grow without oxygen don't grow there, because bacteria that can grow without oxygen could also grow inside of us, so it could be dangerous. And the temperature that we go and grow things at in the school is 25 degrees. So a culture medium used for growing microorganisms might contain various nutrients. So some of the nutrients that is important that it includes is glucose. And if we are going to go and summarise how we carry out this, some of the main things that we need to talk about is using a Bunsen burner to kill any of the bacteria in the air where we're working with a Petri dish underneath the Bunsen burner, using sterilised equipment, spreading the bacteria around on the culture plate, sealing it with a lid and taping it in 4 places, and then incubating the plate for 3 days at 25 degrees. Thanks for watching.